Cause you know I'm all about that bass treble. I'm all about that bass no trouble.
Helicase is an enzyme that unzips the DNA by breaking apart the hydrogen bonds between base pairs and separates it into two different strands of DNA. Next, single-stranded binding proteins comes and binds to the single strands of DNA to prevent the strands from reforming the hydrogen bonds. Then an enzyme called topoisomerase works to prevent tension from building up in the unreplicated portion of DNA. This is only true for eukaryotes. For prokaryotes, the same work is done by an enzyme called gyrase. Now the DNA has divided into two strands called the parent strands. This forms a replication bubble. The area at which the replication bubble is formed is called the replication fork. In the next step, primase comes and attaches RNA primer at the 3' end of the parent strand. RNA primers are annealed to DNA to allow for replication to begin. Once the primer is attached, DNA polymerase III begins adding complementary nucleotides. It reads the parent strand 3' to 5' but synthesizes DNA in the 5' to 3' direction, beginning at the 3-hydroxide of the RNA primer. There are a total of two new strands being formed, leading strand and lagging strand. Leading strand is the strand that is built towards the fork, and it is also synthesized continuously. The lagging strand, on the other hand, is built away from the fork and is also synthesized discontinuously and requires many primers to be synthesized. The process for the lagging strand is quite different than that of the leading strand. In this case, primase comes and attaches more than one RNA primers at various locations on the parent strand. So once again, the DNA polymerase III begins adding complementary nucleotides. It reads the parent strand 3' to 5' and synthesizes in the opposite direction of 5' to 3'. And since this is the lagging strand and it is not synthesized continuously, there are small fragments being formed called the Okazaki fragments. So basically, Okazaki fragments are segments of DNA that are formed from one primer to the next. From this point, DNA polymerase I enters and recognizes the temporary mark on RNA primers. It then removes the primer and replaces it with complementary. This is when an enzyme called the DNA ligase joins the Okazaki fragments on the lagging strand. It joins the fragments by making phosphodiester bonds between the 3' hydroxide and 5' phosphate. So now the DNA replication is complete, and the results are two daughter strands, the leading strand and the lagging strand, which automatically twist into a double helix structure, giving us two strands of DNA.
Here is a piece of DNA. Only one of the two strands of DNA is transcribed. This is called the template strand. The strand that is not transcribed is called the coding strand. This is the promoter region, where the enzyme RNA polymerase is able to bind. More specifically, it contains a data box consisting of a sequence of nucleotides that allow RNA polymerase to bind. RNA polymerase binds to the promoter region and begins to unzip the DNA at its starting point. Here is a more detailed vision of what DNA looks like as RNA polymerase approaches the start point of transcription. Looking more closely, as RNA polymerase unzips the DNA, it forms a transcription bubble. RNA polymerase reads the transcribing strand from 3' to 5', but transcribes newly made RNA from 5' to 3'. In RNA, adenine is complementary to uracil rather than thymine. As RNA polymerase moves forward, mRNA is transcribed and DNA is continuously reannealed. As elongation continues to occur, RNA polymerase eventually reaches a termination sequence, at which point mRNA and RNA polymerase is released from the DNA. mRNA transcription is complete for prokaryotes, but for eukaryotes, a 5' cap is added to the beginning of the 5' section of the mRNA, and a poly A tail is added at the end of the 3' section by the enzyme poly A polymerase. These modifications allow for ribosomal recognition and protection in the cytoplasm. The mRNA still contains introns, which are non-coding regions, or more easily put as useless regions of the mRNA, while the exons are the regions that contain useful coding information. Spliceosomes remove introns and exons are joined together. The introns remain in the nucleus and are degraded or dissolved. The mature mRNA leaves the nucleus and is ready for translation.
Translation occurs when a ribosome locks around a mRNA. The ribosome has two parts, the small subunit and the large subunit. The small subunit locks in place at the methylated cap found at the 5' end of the mRNA. This stage is called initiation. Next, a tRNA binds to the mRNA through the anticodon of the tRNA and the codon of the mRNA. The first codon is primarily AUG. Then, the large subunit of the ribosome binds to the small subunit, creating the P site, also known as peptidyl, and the A site, also known as aminoacyl. The first tRNA is located at the P site, and the second tRNA is located at the A site. Each tRNA anticodon is complementary to the codon on the mRNA.
A certain amino acid is attached to each tRNA. As translation progresses, the tRNA will leave the P ribosome, and the amino acid will bond to the amino acid of the next tRNA through peptide bonds. This creates the peptide chain. This stage is called elongation. tRNA will continue to enter and exit the ribosome until a stop codon is reached. There will be a release factor at the stop codon. This will terminate translation, and the ribosome will separate, releasing the polypeptide tail. The polypeptide tail is a newly created protein. The central dogma theory is about DNA coding for RNA and RNA coding for protein. To take a closer look, DNA creates RNA through transcription. When RNA gets modified, it goes to translation, which is where amino acids bond together to make a strand of protein. There is an analogy where the DNA is like the hardware, and the protein is like the machinery. Just highlighting a few differences between DNA and RNA. RNA is single-stranded. DNA can't leave the nucleus, whereas the RNA can. DNA is like the blueprint, and there are also differences in base pairs.